Hello, everyone. My name is Joe Barco, and I'm Senior Director of Workflow Marketing at Unchained Labs. I will be your moderator, and thank you for joining us today. We will have a Q&A session at the end of the presentation. To ask questions, all you have to do is click on the Q&A in the Zoom navigation bar at the top or bottom of your screen and type your question. We'll get to as many of those as we can. And now I'd like to introduce you to Donna Chen, the Product Manager for Big Tuna. Today, Donna will introduce a combo you didn't think you needed in gene therapy vector development. Big Tuna, Stunner, and Uncle together can help you efficiently characterize AAV constructs and formulations. And now I'll hand it over to Donna. Good morning, Donna.
Good morning. Thanks, Joe, for the introduction.
Sure.
Yeah, I'm Donna. I'm going to be here to tell you about how Big Tuna, Stunner, and Uncle can help you accelerate your AAV stability characterization. Welcome, everyone. Before I begin. Gene therapy is making big waves in the therapeutics world. It promises to cure hereditary disease and rare cancer types rather than just ameliorate the symptom. Take Zolgensma here, for example. It's an AAV gene therapy product. It targets for spinal muscular atrophy by replacing the congenital defective SMN1 gene with functional SMN by using AAV to target the motor core neurons to cure the SMA in one single infusion. Since AAV gene therapy is relatively new, the development process and also characterization methods are under exploration. But the important questions about AAV titers, MT4 ratios, and stability are still important questions to answer, because they all directly impact the capsid design, formulation, and process conditions. But to answer these questions, it takes a lot of time and also precious AAV samples. So I'm going to be here to tell you about the 3 innovative products that we have to help you automate and accelerate AAV characterization. So first we have the Big Tuna. It's a fully automated buffer exchange system to help you buffer exchange and concentrate the sample into the right working range. And then we have the Stunner to quickly give you AAV titer and capsid full empty ratios. And then we have the versatile stability assessment platform Uncle to answer the 2 big questions of capsid stability and also genome injection in aggregation. So we know buffer exchange is a pain. It's labor intensive and it takes a lot of time and hard to manage, especially if you have many samples. That's why we created Big Tuna. You can exchange up to 96 samples in one single run. And we designed the platform to be so easy to use that all you need is 30 minutes of hands-on time from designing the experiment all the way to load the deck and hit the go. And you can come back to finish run. And Big Tuna has been working in many of the protein therapeutics labs, but we also confirmed that Big Tuna can work with AAVs and different viral vectors. And we also know that Big Tuna can work with different gene therapy molecules such as LNP, nucleic acids, and VLPs. So what Big Tuna is a filtration plate-based buffer exchange system. It uses 2 types of SBS standard format filtration plates to do the buffer exchange. We have the unfiltered 96 and then unfiltered 24. So unfiltered 96 does 96 exchanges all in the same time, all in one run, and we can have sample range from 100 microliter all the way to 450 microliter. And unfiltered 24, the volume range starts from 450 microliter all the way to 8 ml. Both plate types use regenerated cellulose as membrane. And if you compare the cost of running single dialysis cassettes or centrifugal units, using the unfiltered is very low cost in comparison. So how does Big Tuna automate the buffer exchange process? So essentially, the method we use is an ultrafiltration and diafiltration method in combination with 3 unique components that helps to automate the system. We have a pressure chamber, an acoustic volume sensor, and gentle orbital mixing throughout the process. So when you start with the sample in the unfiltered plate, first thing Big Tuna does is that it will measure the volume with acoustic sensor first to know what the initial volume is. And it's going to apply pressure to remove filtrate. And after some time of pressurization, it's going to measure the volume again to know how much volume has been removed, and then replenish it with a new buffer. And this will go in cycle iterations until all buffers are replaced with a new buffer. And then you have the option to concentrate the sample if you want to. And all throughout the filtration process, we have gentle orbital mixing throughout. This will increase the flow rate for the buffer exchange and filtration because it's constantly stirring up the molecules to help the filtration go through faster to prevent the samples from aggregating or clogging. And Stunner is actually an ultimate AAV characterization platform. You can run 96 samples at once and get you capsid titer, MT4 ratio, and aggregation information with just 2 microliter of sample. And you can do all this without pre-prep the sample beforehand, such as labeling the sample or run standards side by side. How Stunner works is that you load 2 microliter of sample into the sample well, and the sample will be pulled into microfluidics chamber and be read with dynamic light scattering or DLS, and static light scattering or the SLS, and UV VIS. The combination of the 3 techniques will get you aggregation, capsid titer, and MT4 ratio data. And since all the readings are done with fixed pathways, the result is highly precise and accurate. So what's nice about using the Stunner is that it's going to save you 3 hours running ELISA to get AAV titers. So here we're making a direct comparison of ELISA titers with Stunner titer. In this scatter plot here, we are running 2 types of AAV formats. We have the full AAV9 capsid and also the empty AAV9 capsid. So in the green line here is the full AAV, and the blue is the empty AAV. So ELISA titer is shown on the x-axis, and Stunner titer is shown on the y-axis. So you can see from the scatter plot, the 2 formats of the AAV capsid actually run comparable titer data between Stunner and ELISA, because the slope of the scatter plots on the 2 sample types are close to 1 with a high R-square value. And what's nice about using Stunner to run the ELISA is that you can finish all 64 measurements in 45 minutes. Saves you much time compared to 3 hours ELISA titer. And Stunner also give you accurate full MT4 ratio. So in this experiment here, we're testing the accuracy of the MT4 ratio for Stunner. What we did is we take a known full and empty stock of AAV9 at 2e to the 13th CP per ml concentration, and we titrate them into each other with a known MT4 ratio on the y-axis. So if you look at the 100% full capsid expectation here, the Stunner data shows 100% full. And if we continue to titrate the samples into each other with a known ratio 80, 60 to 40, and you can see the Stunner data is showing that it has great agreement to what the expected MT4 ratio is. And UNCLE is our versatile all-in-one stability assessment platform for biologics, including AAV. It uses 3 different techniques, 3 different detection techniques to give you aggregation, capsid disruption, and also genome injection data. So these techniques include DLS, SLS, and full spectrum fluorescent detection. And besides these detection methods, UNCLE also have thermal control capability that lets you interrogate your sample with different temperature range, starting from 15 to all the way to 95 Celsius degree. And all it takes is 9 microliter of samples to get the stability data. And UNCLE can give you 2 types of stability for AAV. So the fluorescent detection can give you capsid stability by giving you the unfolding information and also give you the genome injection information. And the DLS and the SLS data can give you aggregation at either time 0 or during thermal ramping. Typically, when we assess biologic stabilities, we do thermal stressing on the sample. So UNCLE has a thermal ramping capability together with fluorescence detection capability that can actually give you unfolding information by capturing the intrinsic fluorescent change throughout thermal ramping. So you can watch the protein unfold. And if you add DNA binding dyes such as CyberGold, you can detect the CyberGold signal increase as the DNA is released from the capsid disruption. Besides getting the TM for genome release and also the unfolding information, UNCLE can also give you aggregation information during thermal ramping experiments. So in the TAG here, so by getting the information of TM and TAG in parallel together, UNCLE gives you the full picture of when this DNA is released and when this capsid is aggregated. This is the best way to get you the AV stability behavior in the full picture. So I think of AAV development like a piñatas. So the shell is like pretty much like your capsid, and the candy inside is like your DNA. And the whole process of doing AAV development is all about keeping your piñatas from breaking. And I'm going to take you through an example of how Big Tuna and UNCLE can help with AAV formulation stability screening here. So in this experiment, we take stock concentration, stock AAV9, and then use Big Tuna to exchange it into 5 different common chromatography buffers, and then concentrate the sample 2-fold after the buffer exchange. And then we use the UNCLE to fully characterize the stability of each formulation using the pre-built applications within UNCLE called the TM, TAG and DLS, and viral toolbox. So in this exchange setup, we use the AAV9 with stock concentration of 7e to the 11th CP per ml, and we use the unfiltered 96 at 10 kDa molecular weight cutoff to do the exchange. We exchange it into 5 different formulations with 1 PBS control. That is the AAV in PBS stock, and we're exchanging it into the same PBS formulation. And the other 4 common chromatography formulations also included a highly acidic citric phosphate buffer. We are adding this one in with the intention of knowing that this is not a viable buffer for AAV at all. But Big Tuna was able to buffer exchange and concentrate the sample 2-fold in less than 2 hours successfully. And using UNCLE's DLS application at the time 0 to measure what happens to each of the AAV formulations after the exchange, we can see that the PBS control before and after buffer exchange, that's the green and the blue line here. They are still sized at 25 nanometer, which is the same size as an intact virus, meaning that the buffer exchange process maintain the integrity of the AAV9. And most of the common chromatography buffers listed here also show the same peak at around 25 nanometer. So that's the sodium phosphate, tris, and sodium acetate buffer. But if we focus on the citric phosphate buffer, which is a known harsh buffer for the AAVs, we can see a secondary peak coming up at larger than 100 nanometer in size. This means that this buffer is causing the AAVs to aggregate after the buffer exchange. So using the thermal ramping capability in the UNCO, we can assess the capsid unfolding events in these different formulations. And we can see in most of the buffer we exchange into, they're all having the TM at around 76 to 77 degrees, very similar to each other, with the exception of the citrate phosphate buffer. So it has a lower TM at 71.1, meaning that this buffer is not really, it has a lower TM for this for the AAV, and it's much more readily unfold in this buffer. And during the same thermal ramping experiments, we can actually look at the genome release timing on the AAV. And we're going to focus on citrate phosphate buffer for here, so we can take a closer look on how this buffer is impacting the AAV by comparing it to our PBS control. So in here, we can see the genome is released quite early at around 46 degrees on the citrate buffer in this red line here. And then on the PBS control, it's released much later with a temperature difference of 11.9 degree. And by comparing it in parallel with our melting or unfolding temperature for the PBS and the citrate buffer, we can see that the citrate buffer melted at around 71 degrees, and then the PBS was pretty stable at 76. So comparing these two data together, we can see that the buffer actually have greater impact on the genome release overall, because it has a greater difference in the temperature. Another interesting thing is by comparing the data together in parallel, we can actually see the genome is released earlier, much earlier before than the AAV is unfolded. So UNCO's capability of looking at these data in parallel will enable us to see the AAV stability behavior. If you just look at the thermal melting temperature or the unfolding temperature of the AAV, you will miss that your DNA was actually released earlier. So besides looking at the genome release temperature, UNCO's SLS data will also let you know aggregation timing on same thermal ramping experiments. So in here, the citrus buffer again aggregated very early at 43.5 Celsius, but the PBS control was fairly stable, staying around 74.8 Celsius degree. So by combining all the three data together, you get a full picture of how this virus virus stability behaves. And we have demonstrated that UNCO can show you the size in time zero, and also aggregation, genome release, and thermal unfolding temperature together in one single run with just 9 microliter sample. So what we've done here is that we're able to really accelerate the speed of running all different types of AAV characterization process and really miniaturize the amount of sample we need to run these assays. So by using the Big Tuna to run the buffer exchange and concentration, we're able in the filtration plate format like this, we're able to run flexible volume ranges. And then using the stunner, we're able to run 96 samples and get both titer and MT4 ratio information together with just 2 microliter samples in less than 1 hour. And then using UNCO, we're able to get full stability characterization with just 9 microliter sample using this format that allows you to run 48 samples at once. So here's our recommendation to automate and really fast track your AAV characterization process. You can use Big Tuna to automate your buffer exchange and concentration process and really scale up your sample preparation to do up to 96 samples at once and have the flexible volume range between 100 microliter to 8 ml. And you can use Stunner to quickly give you AAV titer and MT4 ratio characterization information within the same run, which is 2 microliter of sample in less than 1 hour time. And you can use Uncle to get you full stability informations, including aggregation, capsid disruption, and also genome release information with just 9 microliter of sample and in any formulation and in any temperature. So we know you will be able to find the best solution for AAV characterization with Big Tuna, Stunner, and Uncle. Thanks for listening. And now we're open for any Q&As.
Thanks, Donna. So thank you for walking us through these instruments. I see there's a lot of synergy between these 3 instruments being used together. So we have some great questions that have been already been submitted. You can still ask a question by entering it in the Q&A section of your Zoom navigation bar. Joining Donna today, we have Kevin Lance, who's the marketing manager for the Unchained Labs analytical portfolio, to help us answer some questions on Stunner and Uncle. Hey, Kevin.
Hey, Joe.
All right, so it looks like the first couple of questions are on Stunner, so it's to you. So first one is, I'm working with a recombinant AAV. Will that still work on Stunner?
Yeah, that should be no problem. As part of what Stunner needs to run a sample, it typically will take in the amino acid sequence, or if you're working with a naturally occurring serotype, you just tell it what that serotype is. And then from that, it'll calculate everything else it needs to know.
Okay, thanks. Next question is also on Stunner, which is this better for upstream or downstream samples?
Yeah, good question. Typically, Stunner fits in very nicely downstream because that's where you see concentrations in the range that Stunner works the best with. But then its component VIS and DLS can really be used with a lot of samples. But for those really nice capsid titer results, you're going to want the purified samples that you typically see in the downstream processing region.
Okay, thanks, Kevin. It looks like a couple of Big Tuna questions. So Donna, can Big Tuna buffer exchange other gene therapy samples such as LNP, lipid nanoparticles as well?
Yeah, that's affirmative. We have tried LNP, VLP, and nucleic acids with no problem.
Okay. And then what is the AAV recovery after buffer exchange?
So in this experiment, we're able to recover more than 99%.
Okay. All right, and then it looks like some Uncle questions. So Kevin, do different serotypes have different thermal stability?
Yes, definitely. Not only do different serotypes have different thermal stability, it can range across about 20 degrees Celsius, and it also depends on buffer. And that's just if you're talking about the capsid stability. When you start talking about the genome ejection, it's again a totally different ballgame where things can happen 30 degrees Celsius before capsids fall apart, or even simultaneously with when capsids fall apart. So it's really something important to go out and test.
Okay. And then could you contrast, Kevin, the difference between Stunner and Uncle for AAV characterization? So is there one use case or stage that you use one versus the other?
Yeah, it's a good question too. So for Stunner, we get kind of everyday answers. So if I have my sample and I'm going to dilute it and I need to know what's my target dilution range, I want that answer right away. Then Stunner is going to give you a really good answer on capsid titer in 45 seconds. So you can make decisions right away. So you're going to be using Stunner kind of every day in the lab. Uncle is there for answering the really big questions. So when you have to design your capsid for optimum stability, or when you need to pick out your best formulation buffer, or optimize your process conditions for yield, then Uncle is going to help you define where your really big answers are lying. So that's kind of how I think about them. One's really everyday, short, quick answers. The other one's really giving you a nice biophysical readout for complex questions.
Okay, thanks. And then one more question, it looks like, which is it possible to measure in-process samples with residual host cell protein and DNA?
So it gets a little bit trickier then, because you can measure it on Stunner. So I'll answer this for Stunner and for Uncle since the question wasn't specific. On Stunner, you can measure it, but the more non-viral protein and non-viral DNA that you have in your sample, then it just kind of clouds up and interferes with the results. So I would say the best answer is to do this usually after some kind of UFDF step where you've cleaned up a lot of that residual gunk, and then it just shines after you start doing the chromatography steps in your purification process. That's when the results get really amazing. And then on Uncle, especially for the genome ejection behavior, because that's so specific to a dye binding to DNA release, that residual host cell proteins don't really have much of an impact. They will have some impact just from kind of extra noise. But since the dye is specific to DNA, as long as you run a benzonase and are satisfied that you've chewed up all external DNA and you're just left with your DNase-resistant particles, then you can get pretty nice results on Uncle as well.
Okay. Thanks, Kevin. Thanks for your help answering questions today. And Donna, thank you as well. And thank you for a great presentation. So in the audience, thank you for joining us live today. If you'd like to have a deeper conversation with us, please do get in touch with our team. So either your account manager directly, or if you don't know your account manager, info@unchainedlabs.com, or visit the website at www.unchainedlabs.com, which has more information on Stunner, Uncle, and Big Tuna. We'd love to connect with you. Thank you for attending our virtual event, and I hope you have a great rest of your day.